free. And let's dive into today's session. So let me introduce today's presenter. So we have with us today Dr. Jason Stockwell. Jason is an aquatic ecologist who's worked in freshwater and marine sciences from the smallest ponds to the largest lakes in the world. He's a professor at the University of Vermont and also the director of the Rubenstein Ecosystem Science Laboratory on Lake Champlain. If you have visited ECHO, the Rubenstein lab actually is part of that building. So we share that building with ECHO. Jason's research focuses on food webs and how environmental change may alter relationships between predators and prey. Before joining the University of Vermont team, Dr. Stockwell was the station chief at the USGS Lake Superior Biological Station in Ashland, Wisconsin, and was also a pelagic ecologist at the Gulf of Maine Research Institute in Portland, Maine. So for today's session, squeezing the middle of Lake Champlain's food web. The recent surge in natural production by lake trout is a success story, but can too much success be a bad thing? Today, Dr. Stockwell will share new research that he is exploring, the interactions of lake trout natural reproduction and the lake trout stocking strategies to evaluate if too many lake trout mouths will be too much for prey fish populations, especially with the threat of quagga mussel invasion, which could further alter food web energy and food production. So without further ado, I'll turn the screen over to Jason, and we will begin hearing a little bit about that research. So thanks for being with us today, Jason. We're so excited.
Yeah, thank you for having me in the wonderful introduction. Just gonna figure out my screen here. Okay, can you see my my screen?
That looks great.
Okay, great. Well, hello everyone. Welcome to Vermont, if you're not from here right now. So what I'm going to do is talk, as Ashley said, about Lake Champlain, lake trout, some management implications of potential invasive species by quagga mussels and by lake trout resurgence. Actually, a lot of the talk is going to be focused on a macroinvertebrate in the lake that could be key to all of this. And I want to acknowledge my co-authors who are all playing major pieces of the puzzle that I'm going to present today. Drs. Rosalie Bruel, Dr. Aria Chiapella, and Professor Ellen Marsden. And they are, as I mentioned, integral to this effort. In our funding, we're getting funding from the Great Lakes Fishery Commission, Lake Champlain Sea Grant, to do this work, all this work. So I'm going to be talking about the food web in Lake Champlain, and basically starting out, we're really interested in how the further potential invasive species, along with climate change, may be impacting the Lake Champlain ecosystem. And we're going about it in a couple of different ways, which I will elaborate on, including going and doing field observations, and also using ecological models to do some scenario planning, forecasting, evaluation. A lot of this work is both in terms of applied context, meaning how can we use this information to in real world applications, for example, fisheries management, but also a lot of its basic research, meaning curiosity driven. How do we learn about how systems work? And the two approaches hopefully complement each other very well. And so our three main objectives are to basically look at the food web and to connect the dots, so to speak, and find out who's eating who and how energy is transferred among the different trophic levels or the different community members. We want to use models to explore these interactions and to take advantage of long-term data sets that help us train the model and allow us to ask questions, what if scenarios with the models, and hopefully the models combined with the data we collect from the lake and the observations will help us evaluate different potential management scenarios under different future conditions that may help provide insights on the potential challenges, constraints, and opportunities that fisheries management agencies may have in Lake Champlain. So just a quick history of Lake Champlain. For those of you who don't know, back in the 1800s, when the region was settled, there was widespread deforestation, which led to a lot of sedimentation. Lake Champlain has a very large watershed area compared to the surface area of the lake, which means what happens in the watershed really can affect the lake drastically. By the 1850s, there were early signs of eutrophication, meaning increased productivity typically from nutrient runoff. And the lake was also fragmented in many ways. You can see this picture here of the causeway separating Mallets Bay from the main lake. This is about 5 kilometers long. So the lake used to be connected with a 5-kilometer opening, and it's down to about 200 meters through 2 breaks in the causeway here. We've had a lot of invasive species, which I'll talk a little bit more about in a couple slides, but here are some of the more recent big ones: white perch in the 1980s, zebra mussels in the early 90s. Alewife came in the early 2000s and became very abundant by the mid 2000s. And then more recently, we had the spiny water flea. And really recently, we had the fishhook water flea 2 years ago. We've also had problems, as most of you probably know, with cyanobacteria blooms, which really got kicked off in the late 1990s. Attention to this was extreme, or brought to light when 2 dogs died in these 2 years after drinking water from blooms in the lake. This all sounds very doom and gloom. It's not all, because a couple years ago, Professor Marsden's work, she discovered that we're actually getting, for the first time in decades, that actually lake trout were not only reproducing, but they were actually surviving to an older age, which had not been observed before. So this is kind of a ray of hope in usually a gloomy subject of invasive species. And so here's a graph showing the number of non-native and invasive species in Lake Champlain. Invasive species are non-native, but they just have a bigger negative impact. And you can see how we've kind of gone on this extreme uphill increase in numbers of these non-native species. Zebra mussels I'm pointing out here because they're going to kind of play an interesting role in the story I'm telling, because of their cousin, or their specific congeneric, the quagga mussel, which is closely related to zebra mussels. And this is not here yet in the Lake Champlain Basin, but a lot of our work on Lake Champlain food web is revolving around the potential for the quagga mussels to come in. And the reason why we're concerned about that is that even though zebra mussels have really, in the big picture, had relatively little impact as far as we know on Lake Champlain ecosystem, the quagga mussels may be a different story. And I've got some information from the Great Lakes to share about that. So this is a map of the zebra mussel spread in Lake Michigan. In the mid-1990s, you see the darker blue is the higher densities, and you can see how it spread. By 2000, it was surrounding the perimeter of the lake. And then the numbers started going down, and then it's really, in 2010, there's really nothing there. And you say, well, what a great success story. Something happened and the zebra mussels came in and then they left. Well, the problem was that the quagga mussels came in. And what you can see is the quagga mussels slowly or quickly, in 10 years' time, spread throughout most of the lake in very high densities. And you can see it's not limited to just the perimeter like the zebra mussels were, but they filled up the whole lake. And that's because they can colonize much deeper water. They can outcompete zebra mussels, and they have much stronger filtering capacity. So they're a much more a larger threat to the Great Lakes and consequently to Lake Champlain. And so, as I mentioned before, in the midst of this, we've had wild lake trout recruitment. Recruitment is just essentially when young lake trout, the eggs hatch out and the young survive up to a certain size where their mortality stops increasing or is not as high. And so usually when you say a fish recruits to a population, it means that it's reached a stage where the mortality is low. So recruiting to adulthood is good because then those adults will spawn and hopefully produce, sustain the population. So as I mentioned, Professor Marsden discovered this in 2015, but the fish were a couple years old, which meant that the fish were actually surviving since about 2012. And this is after about over 40 years of stocking lake trout and Lake Champlain. Started in 1973. You can see the numbers here, high stocking numbers up into the mid 1990s, and then it declined and has been pretty steady ever since. Again, through all this time period, all the lake trout that you would see or catch in the lake were hatchery origin, were stocked, and not until 2012 have we seen this really tremendous increase in naturally reproduced lake trout. So right now on the lake, there's both hatchery lake trout, and on top of that, naturally reproduced lake trout. So how do you feed lake trout that are in the lake? They need food, they need fish to eat. In Lake Champlain, the native food is rainbow smelt. And the more recent invasive species, alewife, since the early 2000s is here. So lake trout feed on both of these. Very important part of their diet from age 1 or 2 all the way through their life. You can see that they have a negative impact. Because they feed on them, they have a negative impact. But also we're predicting, and based on observations in the Great Lakes, that if we have high abundances of quagga mussels, they filter a lot of the water columns, so they're taking a lot of the nutrients in the phytoplankton out of the water column, which support the zooplankton, which support the prey fish here. And so if these invertebrates, these benthic invertebrates, filter feeders are sucking out the nutrients from the water column, it may be great for water clarity, but it's not great if you're a fish that relies on water column production. And again, I'll talk a little bit more about that as we go on. So forage fish are the crux of the fishery, lake trout fishery. And so our concern is that now, because we not only have stocking of lake trout, but we have natural reproduction, fishery management agencies could control how many lake trout, so how many mouths are out there feeding on forage fish. But with natural reproduction, that control no longer exists. So we have additional mouths out there. And the concern is that maybe if there's too many mouths, they'll suppress the prey fish population to the detriment. But if we also have quagga mussels that are sucking the productivity out of the water column, then that might squeeze the food source for these prey fish. So they may be squeezed through predation effects from lake trout, and they may be squeezed from the bottom by nutrient depletion from these zebra mussels or quagga mussels filtering the water columns. And so our question is, if that's the case, how could management agencies adapt to these conditions? And so a central question we're asking is, where does the energy for the forage fish come from? Does it come from the water column, or does it come from the bottom or the benthic zone? Benthic means bottom, pelagic means water column. And that has implications if we have a quagga mussel invasion and the quagga mussels suck most of the productivity out of the water column and bring it down to the benthic zone, where these fish get their energy from will have implications for whether they do well or not do well in the presence of quagga mussels. So we know that smelt and alewife eat zooplankton, and this is a picture of Daphnia, which eat phytoplankton. So this is all kind of pelagic resources. But we also know that smelt and alewife also feed on mysis. So mysis are really interesting. They're a macroinvertebrate. I'll talk a lot about them actually in the next couple slides, but they're omnivorous, which means they can eat the bottom mud, the bottom invertebrates, but they also can come up at night and eat plankton, zooplankton, and phytoplankton because they exhibit this DVM, diel vertical migration, which means they come up at night and they go down during the day. So they play a really important role in the food web because they are large, relatively speaking, a large energy packet of food. They can migrate from over 400 meters depth in deep lakes all the way up to near the surface in a single night and go back down. They're about an inch long. They like low light, so they'll only come up at night typically near the surface, and they prefer cold water, which they need to because they live down deep. And because they're relatively large and they go up and down the water column, fish love to eat them. They're a big energy packet. They're full of lipids. And so pelagic fish such as smelt and alewife will feed them. Bottom fish such as sculpin that live on the bottom all the time, they'll feed on them when they're on the bottom. And then some fish, this is called a quagga from Lake Superior, they will actually migrate with the mysis, and over 90% of their diet typically is mysis. So they're important for the fish community in all parts of the water column. But they're also important because, as I mentioned, they're omnivorous. So they'll not only eat zooplankton and phytoplankton up in the pelagic zone, they'll also eat benthic invertebrates, so invertebrates that are on the bottom, and they can also eat detritus or decomposing material. Think of phytoplankton settling to the bottom and decomposing. Mysis will eat the mud essentially. And so they're not only feeding the fish community, they're impacting these other parts of the food web, and their movement is bringing energy and nutrients up and down the water column to all different parts of the ecosystem. So really important players. And we've done some studies on them to demonstrate that. This is a food web that we constructed from Lake Superior in the mid-2000s when I worked in Wisconsin. And basically, here's mysis right here. Here's kind of these other fish, smaller fish species such as smelt. And there's no alewife in Lake Superior, but there's sculpin. There's these things called bloaters and ciscos, and then there's these predatory fish. And what you'll notice is that mysis have a lot of arrows and a lot of thick arrows coming out of their box, which means that most of the fish community feeds on them, depending or regardless of whether they're pelagic fish or benthic fish. So whether they're up in the water column or they're on the bottom. So they're very important to fish. They're also, we know they're important to food webs because of their impacts in certain systems. So this is Flathead Lake in Montana. This top panel was the original community, which had very few fish species, zooplankton. And then sometime in the 1900s, they introduced kokanee salmon and some whitefish, and some lake trout got in there. And the kokanee did really well. They fed on the zooplankton. It was pretty good for fishing. But then they introduced mysis. And what mysis did was that they actually consumed all the zooplankton, and the kokanee could not eat the mysis because the mysis would go down during the day and up at night, and the kokanee didn't overlap with them and they couldn't see them in the dark. But the lake whitefish and lake trout, which are bottom fish, they all of a sudden had an energy source to feed on. So those populations started taking off. So this small macroinvertebrate about an inch long can have really strong ecosystem-wide impacts on lake systems, which makes them really fascinating. And so this behavior, this diel-vertical migration, DVM, is really key to a lot of this. And so our understanding of DVM is that during the day, the mysis stay at the bottom all day long, and then at night they kind of come off the bottom and they feed on the plankton, all the plankton up here. So their reward, it's hot, there's a lot of plankton, these hot colors, and there's very little plankton down deep. So the idea is they go down deep to hide from the fish predators, and then they come up under the cover of darkness and feed. The risk is very high during the daytime because of the light, but at night the risk is very low because of the dark. And for this reason, many people who work on mysis, who sample mysis, they work at night under red light, as shown here, because the mysis can't see red light, so the boats don't scare them away. Most of the sampling occurs in the water column because it's easier to sample than on the bottom. And the assumption is that that most of the population up here in the water column is is representative of the entire mysis population. But when we look at the research literature, for those few studies where they actually sampled mysis both up here in the water column at night, but also on the bottom, what they found is that mysis are found on the bottom at night also. Up to 85% of the mysis caught were actually on the bottom rather than up in the water column. And so that suggests an alternative hypothesis to their behavior, that not only at the bottom during the day and come up at night, but they also remain on the bottom at night also. And what this suggests to us potentially is that maybe the bottom might be a better environment for them than we thought. And our idea is that maybe all the stuff on the bottom, the detritus, the mud, the invertebrates, might have some valuable or more valuable reward to the mysis for consuming than we previously thought. And so maybe they don't need to migrate. And so this is called partial migration, partial DVM, where the population consists of mysis that migrate and mysis that do not migrate. So residents and migrators. And this is actually very common in the animal kingdom. A quick search around the literature shows that all these species and many more actually are partial migrants. So you can think of geese flying south in the wintertime for the winter, but actually some of them actually stay up here. So we have migrators and residents, and that's the case for here. So we're starting to think of mysis behaving similarly to these other animals, just at different spatial and time scales. So daily versus seasonally, and going 100 meters instead of hundreds of kilometers or thousands of kilometers. So we wanted to ask the question, do mysis actually do partial migration in Lake Champlain? And if they do, to what degree? And so we went out and we sampled in the pelagic zone with these nets that were just in the water column. We sampled on the bottom with the sled. There's some rudders here that we drag this along the bottom and collect them in the net. And we also deployed video cameras on the bottom that look down to look at what's going on right on the bottom of the seafloor for 24 hours at a time or more. The lake floor, not the seafloor. And we sampled day and night each month in 2015 at 2 sites in Lake Champlain, a 60-meter site or 180 feet, and a 100-meter site or 300 feet. And that was located off Burlington. Burlington's right over here. Here's Shelburne Bay. And we used all these different techniques to sample. And so what we found was that at our 60-meter site, the shallow site, at night, indicated here, this is mysis density in number per square meter, and this is the different months. What we found at the 60-meter site in the pelagic zone or in the water column, we only caught mysis at night and didn't really catch any during the day, which is what you would expect under diurnal migration. And on the bottom at night or during the day, we caught them on the bottom during the day as we would expect under vertical migration. They stay on the bottom during the day and come up at night, but we also caught them at night on the bottom. Now, when we looked at the 100-meter site, so that supports this idea that they're on the bottom during the day, but at night they come up, but some of them stay on the bottom. At the 100-meter site, in the pelagic zone, we catch them at night as we expected, but we also caught them during the day as we did not expect under that DVM. And at night, or in the benthic zone, we caught them both day as we expected, but also at night. And so what this suggests is we have mysis not only on the bottom during the day, but in the water column during the day. And at night we have them in the water column, but we also have them on the bottom. So this is kind of, in terms of mysis ecology, this is really interesting that they're exhibiting all these different types of behaviors. So it supports that alternative hypothesis that they are undergoing partial diurnal migration. In our video camera, here's a picture on the bottom. All these dots are individual mysis. These 2 lights are 2 lasers that are going down. They're about 10 centimeters apart. And what you can see is that at 12 noon on this day, all these mysis were on the bottom. We could count them, and we know this area, so we can get a density estimate. And you can actually see that they're all oriented in the same way because there's a card on the bottom. And what we can see is that in all these graphs, we have the number of mysis per square meter over time of day from 8 a.m. to the following morning, and then into the following day and evening. And the shaded areas are night, and the white areas are day. And you can see the phase of the moon here. These are new moons, and this is half moons. What we would expect to see is that there should be a lot of mysis on the bottom during the day and very few on the night. And what our cameras say is it's highly variable. We see them on the bottom during the day, but we actually see higher densities on this day at night, and then lower densities during the day. We have different patterns that they're there on the bottom at night, but they increase during the day. 2 different years, we had slightly different behaviors going on at the end of the night where the densities went down, but the densities increased. So it's really variable from this is showing us some really interesting behavior that we wouldn't see with our normal equipment. And again, it supports that things are highly variable and there's partial migration going on. One thing I want to point out is look at these densities. They go up to 2,500 in a square meter. So if you think of a square that's about as wide as your arms are, there can be up to 2,500 of these mysis in that area. Really dense. And what we thought this was really interesting because when we compared it to our benthic sled estimates, that sled we tow on the bottom and catch them in nets, our maximum observations were only 50 per square meter. So what this suggests is that we may be missing a lot more on the bottom by our traditional methods than what we actually see when things are undisturbed. So there may be a lot more of them out there than we thought. And so again, this information suggests that partial diurnal migration is the rule rather than the exception. So when we look at the literature on mysis research, what we find is that all these are different measurements that people do on mysis. So how many are there? How much weight or biomass of them are there out there? What's their average size? How fast do they grow? If you look at all the information in the research literature, most of the information is taken from studies that only sampled the pelagic environment. So all this, so 75 to 80% of the studies only look at the pelagic environment. Very few studies look at the bottom, and very few studies look at both habitats. And what we're saying is that we should be looking at both habitats because we find mysis in both environments. So we may be missing information on mysis, and I'll tell you why this is important. Oh, before we go on, we have our first poll question. Ashley, are you there?
I'm here, and so I am just launching that poll, so you should see that popping up on your screen. And so the first question asks, mysis are important in lake ecosystems because A, they move around, or they move energy and nutrients between the surface and the bottom of lakes. B, fish that live in the water column eat them. C, fish that live on the bottom eat them. Or D, all of the above. And so if you do not see that chat box or the polling question popping up, go ahead and just put your answer right in the chat box. It looks like some answers are rolling in here. That is phenomenal. So again, think about, why are mysis important in lake ecosystems? Are they A, moving energy and nutrients? B, are they getting eaten up by fish that are in the water column? C, are they getting eaten up by things that are at the bottom? Or D, all of the above? All right, I'm going to go ahead and share our results from this poll. And Jason, talk us through this.
Okay, so there really are no wrong answers here, but probably the most correct one is all of the above. Because they move up and down the water column and they're eating things in both places, the bottom mud, the bottom invertebrates, but also the plankton that are up. And because they're moving, they're converting all that energy into their body, and they're moving up and down. They're excreting nutrients in both places, so they're moving energy up and down. And then they're also, because they're converting that energy into their own body mass and fish can eat them, whether they're up in the water column or down, it means it just means all of the above, that everything's eating them and they're moving energy up and down. That's what makes them really important to ecosystems. Okay, so let's move on. So the question, to get back to our question originally, where does energy for forage fish come from? We have this question of, are they eating plankton, pelagic resources, or are they eating more benthic resources? And the mysis play an important role because the mysis consume benthic resources and then move up in the water column where the forage fish can eat them. And so we want to know, especially in the case of how might this impact them, the energy sources, because the quagga mussels might be able to suck out energy from the pelagic resources to the bottom. So here's a kind of a pictorial view of our hypothesis. So here we have alewife and rainbow smelt in a higher trophic position, meaning they're more predatory than the zooplankton, which are predatory on phytoplankton. So think of this as a food chain increasing in this direction. And then think of the bottom of the lake in the water column of the lake. And so if alewife and smelt are consuming both zooplankton and mysis in equal parts, but mysis are getting more of the diet from the benthic zone because they're eating more of it compared to the pelagic zone, then this might suggest that alewife and smelt might be resistant to quagga mussels, because we expect the quagga mussels to suck energy down to the bottom, where mysis can still take advantage of them, convert it, and still move up and have rainbow smelt and eat them. So in this case, if this was the case, then we might expect less impacts of a quagga mussel on alewife and rainbow smelt, which means they might not be squeezed from the bottom as much. The alternative is that if they're eating zooplankton and mysis are getting more of the energy from the pelagic resource, if quaggas come in and suck out the energy from the pelagic zone, then alewife and rainbow smelt may be more or less resistant, but more susceptible to being squeezed from the bottom because not as much energy will be available to them. And so the way we're evaluating this, we're going about this in 3 different ways. We're going to look at stomach contents of the fish, and we're also going to look at the fatty acids of the fish for reasons I'll get into. And also we're looking at chemical things called stable isotopes, which basically are chemical tracers that can help us map out the food webs. We do field collections for all the tissue samples we need, along with the stomach contents. We have a lot of students involved. All the people pictured here are undergraduate students or graduate students. And so let's talk about stable isotopes. Basically, it's a chemical signature to estimate diets of fish and other things over time. Here we have carbon isotope on this axis, and basically if the ratio of these different carbon isotopes are low, that signifies things that are produced with pelagic carbon. If it's higher, that signifies things that are produced more on the bottom or in the nearshore zone. And basically, if you eat more of these, then your signature is going to be more on this side of the carbon scale. If you eat more of these, your signature is going to be on this side. We can also look at nitrogen, the ratio of these isotopes. And as this ratio gets higher, you can tell trophic position. So zooplankton will have a higher nitrogen signal than phytoplankton, and fish will have it higher than zooplankton. So basically, if you can measure the chemistry of fish and of these other parts of the food web, you can get an estimate of where the carbon is coming from. So let's say if a fish was eating a 50% zooplankton and 50% benthic invertebrates, and the benthic invertebrates ate all carbon from the benthic zone, and the zooplankton ate all carbon from the pelagic zone, if you put these together in mathematical models, it will basically give you a carbon signature that is in between the pelagic source and the benthic source. Now this, I think you can probably see how this would be really helpful, but the problem or the challenge is that mysis eats things in both places. And so where mysis are eating and then how much of the diet of fish is mysis will affect their signature. And so from that, so by looking at the isotopes of the food web, we should be able to tell where the carbon is coming from, pelagic or benthic resources. Similarly, fatty acids can also provide us information on their diets because they can act as natural tracers in the sense of you are what you eat. There's a big push, you know, a healthy diet includes omega-3s. Omega-3s happen to be produced by phytoplankton, which if zooplankton eat them, they're rich in omega-3s. Again, in the bottom and side habitats or shoreline habitats, omega-6s are more important or produce more. And again, this is transferred up the food web. So depending on how much a fish is eating of each of these sources, you can measure its fatty acid composition. And if it's eating more pelagic resources, it would be high in omega-3s. And if it was eating more benthic resources or littoral resources, it would be higher in omega-6s. So again, measuring the fatty acids would tell us something about where the energy is coming from. So as I mentioned, this scenario, forage fish could be squeezed from the top or the bottom. We kind of went from the bottom here, but again, if lake trout are resurging and stocking is continued and there's naturally reproduced lake trout on top of that, there's many more mouths that are eating forage fish. So the question is, if we adjust lake trout stocking rates, might this relieve the pressure on the forage fish, especially if they're being squeezed from the bottom? So this, to look at this, we're doing modeling. Again, this is to hopefully look at how the food web is put together and then start playing games or playing scenarios of what if scenarios. And this is a lot like making decisions. This is from the trolley car, classic example. The trolley car, if it's going down one railway and it's going to hit somebody, but you have a chance to divert it. But on this pathway, there's 5 people. Which one are you going to let it go and run over one person or 5 people? There's all sorts of ethical, philosophical questions about this. Well, what if the one person is going to do something great and beneficial for the world, and these other 5 people are maybe bank robbers? And so how you make your decision has implications. And so we think of modeling scenarios, in this case, management decisions. So we could go business as usual. We could make a different choice and go down to scenario 1, and we can do a different scenario 2. And not only do these have different repercussions of, say, the food web, but there's also different financial outcomes too with that. So models are really good for playing these sorts of games. And so we're using this model called EcoPath, which was developed for marine systems. Again, this should hopefully look familiar with arrows, different parts of the food web, who's feeding on who and by how much. We're essentially going to plug this model into our lake data for Lake Champlain and start doing different scenarios. And so Lake Champlain is really great because there's a wealth of information over the long term. The state lake monitoring programs for phytoplankton and nutrients and temperature and zooplankton are wonderful resources. The state has also been monitoring the fish such as smelt and now alewife. We know how many fish have been stocked into the lake. So there's all this information that can be kind of fill in those food web compartments. And then if we know something about who eats who and how much and what the energy conversion is, we can start tweaking the food web model. So this is a preliminary model that we've made. Rosalie's been doing this. And you can see here all the different food web compartments numbered. And so if we look at mysids here, number 15, that's mysids. You can see there's a lot of arrows coming in and a lot of arrows going out. Again, that's their important part of the food web. But we can also look at the relationship for stock lake trout and wild juvenile lake trout, which are 2 and 3, and how these might transfer into lake trout, adult lake trout. And then sea lamprey, which I haven't really talked about, that's another species that feed on large fish. And so we're putting together a food web, and we'll be able to, again, basically do different scenarios. What if we decrease stocking rates? How will that reverberate throughout the food web? What if we were to let me see where I'm looking for I guess we don't have zebra mussels in here. So, but you can think of adding zebra mussels or quagga mussels in here. Probably don't have zebra mussels because they don't have large impact. If we add zebra or quagga mussels in here, all of a sudden they may be sucking down 18 the phytoplankton and contributing more to detritus, so shunting these pathways, so making this arrow bigger and these arrows smaller. So we can do that. And also we haven't included in our modeling yet humans. So humans, we do see lamprey control, so we're decreasing their biomass. And we've also been adding juvenile lake trout into the system. So that's inputting energy at a different level. So the model will be able to help us play these games and evaluate different scenarios. And so for instance, we might want to look at the effects of different stocking practices or the effects of invasion such as quagga mussels, or even looking at the impacts of climate change if it's changing the temperature of the lake or the seasonality of the lake, because temperature is really important for how fast things grow, how fast they consume things, and are related also to mortality. So we can, you can hopefully see how we could play around with the model to ask these questions and basically do different hypotheses. Will different stocking rates have a bigger impact on the food web or on the lake trout population than the effects of, say, quagga mussels, greater than the effects of climate change? Or will climate change have a much bigger impact on either stocking rates or invasive species? Or will the interaction of, say, stocking rates in invasive species have a much larger impact than just climate change alone? Okay. So I have another question here. Ashley, are you there?
I'm here. I'm just getting ready to launch poll number 2. And so this one is, really important. So make sure you think through your answer on this one. So looking at some of the data and information that Jason has shared, he is asking, math and computer skills are important for careers in fish and wildlife. So you've got option A, which is true, option B, which is false, and C, I love math. And again, if you're not seeing that, go ahead and put your response rate in the chat box. That's okay. All right, so let's see. What do people think here, Jason? So it looks like most of you say true, and we've got one person that loves math.
All right. That is great to hear. Yeah, so I'm gonna speak a little bit more about this, but hopefully it was obvious that it's not all about being outdoors. And I got a slide to follow up the poll on the next slide after this. But I want to talk about the skills necessary for this research program, especially for the students out there. So some of the skills you need to do this type of research is you need to be able to go on the field and sample with water bottles, plankton nets, benthic sleds. We trawl for the fish, and being able to, out in the field, to identify the fish. Those are all important field skills. In the laboratory, we need to filter water. We need to sort samples. We need to weigh and measure fish and dissect them for their stomachs. We have to do things with the samples, such as drying them in ovens, which doesn't smell great sometimes. And we have to grind samples. So there's a lot of technical skills in the lab. Often overlooked is you need to be organized to do this work. So there's a lot of data we're recording. There's a lot of planning. So you need to be organized. You need to write things down clearly. To do this all effectively, you really need to understand what you're doing. So understanding why you're doing something's important. Being able to ask questions of why things are going on will make you more involved and take more ownership. And there's always problems that come up. So thinking clearly, asking questions, and being able to come up with solutions are all really important skills that maybe we don't think about. And finally, the quantitative skills, which we just mentioned. So a lot of the things we use Excel quite a bit. So I'm sure a lot of you are using Excel, not only to enter the data, but we need to make sure the data are entered properly. So we do a lot of checks on that. Summarizing the data with basic statistics such as averages are really important. More sophisticated work, there's programming languages that you can actually do statistical analyses at and graphs called R. Summarizing and visualizing the data are all important. So I guess to get back to the poll question and to this, some unsolicited advice. It's very easy to go out and to set nets and to capture fish and to take pictures of fish and to really have a good time. And you see on the right picture, bright sunny day, blue skies. So this is, I would say this is probably 5% of doing research. The harder thing is how you handle all the things you collect out there. So the data analysis, processing the data. This is not as easy to do, it's not as glamorous, but it's far more critical. And so my advice to all the students out there is to pay attention to math and take math courses and computer classes, because it will set you apart when you are looking at careers in natural resources. Again, most everyone can easily learn to do this. It takes a lot of effort and resilience to do this. So with that, that's all I have, and I hope you enjoyed it, and hopefully there are some questions.
Great. Thank you so much, Jason. It's fascinating to think about all of the different interactions that are happening out on Lake Champlain. I think a lot of these pieces are often missed or overlooked because we can't observe them, or we're present during only a part of that timeline. And so thinking about a 24-hour period and what's happening over that duration, there's definitely, a significant bias just as humans to want to be there in the daytime, right? And to observe what's happening during the day. But it's really important to think about when you're designing research and setting projects up, what's happening over kind of a larger time scale. And actually, you just said something the other day that really stuck with me and made me think a little bit more about how we connect with place. It's thinking about, if you really know a place, go there at night and see what's happening and see if that changes your perspective. And so thank you so much for sharing about the work that you and your team are doing, Jason. I think it's so valuable to take some of the quantitative science that you're talking about and put it into these data visualization models so that you can see energy flow. You can model how some of these changes that are kind of coming down the pipeline are going to impact this wonderful resource that we have here in Lake Champlain. And so we've got questions coming into the chat box. We can dive into a couple of those. So Karen wants to know, does excess phosphorus into the lake affect mysids? Do mysids feed on cyanobacteria? And how healthy is the plankton population in Lake Champlain?
Okay, so the phosphorus will generally probably benefit mysis in the sense that it's going to increase primary productivity, which will typically increase zooplankton populations. And so that's beneficial for mysis because they eat that sort of thing. So overall, so that's kind of a bottom-up positive effect. In terms, do mysis eat cyanobacteria? I would, they eat everything. So I would probably say if they encountered it, they would, as long as it wasn't toxic and they knew that. However, mysis require very deep water, so probably minimum of 40 or 50 meters, so 120 feet. And so more often than not, the cyanobacteria blooms are typically more warm water, shallower lakes. So the overlap of mysis with cyanobacteria blooms are not there's not a lot of overlap. If you think of Lake Champlain, a lot of the blooms are in St. Albans Bay, in the South Lake, in Missisquoi Bay. I mean, there are some that are out in the main lake at times, but mostly mysis are offshore, so there's not a lot of opportunities for that interaction. Did I answer all that?
You did, and then there was the kind of last piece of that question was just talking about phytoplankton populations in Lake Champlain in general and kind of their overall health.
I think depends on where you are in the lake. So again, the cyanobacteria blooms in the shallower bays, those are issues. I think out in the main lake, the plankton population is probably what you would expect. We do have Bithotrephes, the spiny water flea and the fishhook flea came in, and those have the potential to alter the community structure by feeding on other plankton that otherwise would be available to fish. So there's probably some competition going on between fish and those fish eat plankton, and the invasive, the waterhook flea, are eating the same plankton, although the alewife and the smelt will eat the spiny water flea and the fishhook flea. So it's we could do another whole model just on those relationships.
Great. And then the next question actually, and this is interesting. So they're asking, mysis are native to Lake Champlain, and they're asking kind of when they showed up. I think this question is asking when they're introduced in Lake Champlain. Maybe you could speak to kind of when research started on mysis, unlike, let's say, Lake Tahoe, where they're invasive.
So in Lake Champlain, so mysis are called the Glacial Relict because they're originally a marine species. And so if you think of the history of Lake Champlain, it was part of the ocean. I think it was at one point a large bay that entered into the St. Lawrence River. So once, so mysis were here then, and so once the lake got cut off from the ocean and converted into salt, or from salt to fresh water, the mysis were already here. So they've been here all along since Lake Champlain formed. In other systems where they've introduced them, typically a lot of introductions were done in the 50s and 60s and 70s. And so I don't know exactly when. I think Tahoe was probably stocked with mysis in the 60s some point. And there's been, depending on the system, there's been research either going on right away to evaluate the effects of those stocking programs, or they were just put in and forgotten about and not evaluated till later. There's quite a few lakes out west that have mysis in them.
Yeah, thank you. That's great. And I actually have a question about that in the terminology you're using. So you're saying that they were stocked intentionally versus a lot of the aquatic invasive species that we deal with today are kind of inadvertent, coming from bait fish or coming from ships and boats that are moving around in water bodies. Is that correct?
Yeah, so the idea was, as I mentioned, mysis are of a sufficient size that they're really good fish food. They're big and they have lipids. So think of a lake, I'll just say out west because that's where a lot of lakes with good trout fishing. Managers got the idea that the trout fishing was good, but if we put mysis in the lake, the fish will get even bigger because they'll have more food to feed on. But they didn't think through it completely, in that this vertical migration behavior, mysis basically came up at night when the trout could not feed on them, and the mysis would feed on the zooplankton, then they'd go down during the day, and the trout would not go down that deep during the day. It was too dark and probably too cold. And so essentially these little macroinvertebrates outcompeted the trout for their own food. And so what happened was those fisheries actually went downhill. The growth rates declined, the numbers of fish declined. And so ironically, mysis were the ruin of a lot of fisheries because of that vertical migratory behavior.
That's great. Yeah, thank you for sharing that. So we have a couple questions in the queue. I'm just looking at the clock here. So what I'm going to do, I'm going to launch our feedback poll. And while you all are voting on that, I'm going to give Jason an opportunity to just answer these last couple of questions that are in our queue here. So you should be seeing the feedback poll popping up. Go ahead and fill that in. It's super helpful for us just to get feedback from you all on how you enjoyed the session. And then I'll just pose these last couple of questions to Jason. So one question is kind of a follow-up question on the spiny water flea, is looking at, is it still too early to predict the impact and effect of the spiny and fishhook water fleas in terms of impacts on food chains, and what kind of results we're currently seeing with salmonoid populations that are feeding on them?
So my understanding, I haven't been tracking. I have not been tracking the population dynamics of the fish with flea and the spiny water flea. SUNY Plattsburgh has a zooplankton monitoring program, Tim Hook, so he probably has a better pulse on it. I don't think it's too early to see an effect because of their plankton nature. They can populate really quickly and have a large predatory effect quick. From what I've heard from our captain the past year, some years we have lots and lots of spiny water fleas out there, and some years they're a very minor component, at least from their regular sampling. Some years they show up early, relatively early in July. Other years they don't show up till later in the fall. So presumably later in the year they show up, the less time they have to impact the system. I'm not right now. Our study is really one of the only ones that are going to be looking at the diets of smelt and alewife, to my knowledge. So we'll hopefully have a better handle on how much the fish are consuming that. The work in Ellen Marsden's lab on young lake trout doesn't suggest they're feeding very much on Bithytrophes or the fish with water flea, and that again might be more of a spatial overlap in time because those fish tend to be more bottom oriented.
That's great, thank you. And then Mara has a question, which I think is probably a lot of people are wondering about this out there. What invasive species have had the biggest impact on Lake Champlain to kind of to this day?
Oh, that's a good question. Well, I guess one could say sea lamprey, although there's some controversy whether sea lamprey are actually invasive or native. And so if you consider them invasive, then they have had a really large impact because of their impacts on top predatory fish, such as lake trout and the salmon that are stocked. As I mentioned, zebra mussels don't seem to have had such a big impact, at least in an ecological sense. I'm probably missing something here. White perch is all over the place in high abundance, but we still have yellow perch and other fish that could have displaced. So I wouldn't say that has had a large impact. Yeah, I guess I'm not the plant.
Kind of depends on what you're looking at, right? And what you're evaluating for. So probably Ellen Marsden was here. She would say lamprey, I'm sure. And I think if you talk to some other folks that were looking at plankton communities and water clarity, they might say zebra mussel.
Yep. You know, people who are focused on aquatic vegetation, milfoil, water chestnut.
Yep. That's great. And so we actually are going to do a session coming up in a month here on aquatic invasives. So Mara, stay tuned for that. I'm going to have, we're going to advance to our take-home activity and get ready to introduce that. But one quick question, rainbow smelt and alewife, are they stocked or are they introduced? Coming from John. Okay.
Rainbow smelt are native. They've always been here. Alewife are not native. They're invasive. And I believe the thinking is that they were transplanted here by somebody.
Awesome. Great. Thanks so much, Jason. So for today's take-home activity for this session, we are asking you to think about drawing a conceptual picture. So a little scientific art here, thinking about what might this mysis migration behavior look like if you were to draw it in kind of 4 chunks over each of our different seasons. So in the winter, summer, spring, and fall. And so we're going to put a link in the chat box for that. And then for our next session, I'll just give you a quick blurb on what we are going to be talking about on Tuesday. So on Tuesday next week, we have Jarlath, who is going to be joining us from the University of Vermont Spatial Analysis Lab. And so Jarlath is going to be talking about mapping streams with drones. And so thinking about how you can actually gather a lot of data in a really quick time frame and use that for floodplain management, for mapping geology and things like that. And so that'll be Tuesday at 12. We will put our link in the chat box for you to join us for that. And without kind of anything else here, because I know we ran over a couple of minutes, I want to thank everyone for joining us, and we'll be so psyched to see you again here next week. Thank you, and thanks again.